Uh, I was attending Human Proteome Organization World Congress in Orlando. Just came this morning. Was very interesting congress. Lot of new innovations in the area of proteomics. And actually, a lot of things are evolving towards how to do validation, how to do targeted assays, how to do more robust computational assays. That's where a lot of emphasis is happening in the area of proteomics field. So today's session is actually very relevant in that way because we are talking to you that how to take your discovery-based findings forward and do validation. So if you remember, we the last 2 weeks we have been discussing about mass spectrometry basics, ABC to XYZ of mass spectrometry, and within them we started with the basic for protein identification. The last week we talked about protein quantification, and today here we are going to discuss about validation strategies. So let me start with some slides. If you just to recall you the previous uh, session which we had in the last 2 weeks, essentially we have been discussing about how to prepare your protein sample, uh, do the protein digestion, then you can get the peptides which you can separate further using liquid chromatography, and then you have to do the mass spectrometry analysis, for which you can do electrospray ionization, followed by different type of mass analyzers. Especially we talked about orbitrap technologies, and but there could be variety of configurations like QTOF or QE orbitrap, or you can have diffusion, which is hybrid platform. So variety of ways you can analyze your sample at MSMS level, generate these mass spectrum, and use those information for B and Y ions to do further subject search for databases, which will tell you the protein of interest. So in this way, you got uh, probably a quick recap of the workflow. Let me again give you a quick another recap of quantitative proteomics workflow which we talked. For example, if you are doing TMT based, tandem mass tag based quantitative proteomics, for which you have let's say 6 conditions to compare. You have added the reporter ions from 126 to 131, and added each one of those reporter ions in your each condition, 6 conditions. And then after doing MSMS analysis, now you are fragmenting your reporter ions, and you can now see those masses can be seen for reporter ions from 126 to 131, and this information could be used for quantification purpose, right? So let me take a pause here and talk to you briefly. Yeah, so far I hope you are now remembering what we discussed in the last 2 weeks about the common workflow for doing mass spectrometry based proteomics work, and then doing the quantitative proteomics using tandem mass tag or iTrack per se. Uh, so then let's say you have identified some proteins of interest or peptides of interest, and you want to do further validation. So what options you have available? The options mainly what you have is you'd like to identify which antibodies are available from literature, from the vendor catalog, and then you'd like to purchase those antibodies to do either Western blot analysis, or you'd like to pursue for the ELISA or some other based scanning based method. So now your choices are very limited because not many proteins uh, have good antibodies available. So you are limited with those antibodies, which you can now take forward for your actual biology experiment. If you are, let's say, fortunate that you got antibodies of interest, you can now do validation and you can move forward with your experiment. But many times your top hits, your the proteins of most interest, you may not have antibodies for them. So how to take them forward? Let me again resume back on my PPT. So this is what we have shown here on the slide. that these are the major two ways of doing the validation which people use popularly, a Western blot or ELISA. Uh, but many times you may not have access to the good antibodies or the reagents or commercially available to do these kind of validation experiments. So then how to take it forward? And that's where the target proteomics field came. And this paper was published in Nature Methods in 2013. At the cover page stating the method of the year, uh, depicts that, in addition to your discovery workflow, from the same sample, same peptide, you can also explore an alternative path, which is targeted proteomics workflow. As the name says, here you are looking at very specific peptide, very target peptide. You are not looking at all the peptides possible. So in the right hand side, you are interested to screen for every peptide present in the complex samples, and you are trying to identify the patterns of those common peptides which are showing some differential expression response in the disease individual as compared to healthy for a context. But if you go on the left hand side, you already now know which peptides to screen, and you are only selecting those peptides, and then you are utilizing a triple quadrupole based mass spec and only selectively looking at what happens to my that peptide and its various transitions. So this is the alternate workflow which came forward, and that is what we are going to talk today in much more detail. So there are a variety of ways of doing the targeted proteomics. Uh, I'll interact with you again briefly. Okay. So now you got an idea that there can be two workflows. One is discovery workflow, and second is the target workflow. In the target workflow, you are selecting the specific peptides, and now you want to do further investigation of those in large number of samples. For example, you want to validate. So there could be many ways of doing this experiment. One standard way, which is started, is known as SRM method or selected reaction monitoring method. But there is an alternative method to that as well, which is known as PRM or parallel reaction monitoring method. People have also come with much more high throughput approaches of screening every possible peptide and doing their quantification, and that is more covered under the SWAT or data independent acquisition DIA method. So let me again share you the slide, which is giving you the schematic of uh, various workflows available. So this slide conveys you the detail which I'm talking to you, that you can look for either the SRM or the various peptides, their transitions, multiple reaction monitoring, or you are doing all possible peptides and their transitions you are monitoring under the PRM. And an alternate method of these two is data independent acquisition, either doing the Orbitrap based technologies directly DIA, or you can use a triple top direct based instrument which can be used for this fast MS based workflow. So for today's hands-on session and the discussion, we are going to focus mainly on SRM and PRM. Of course, there are lot of advantages of using SWAT and DIA as well. Not to underestimate that these two are not good, but these two are still very high throughput approaches, and sometimes also instrument specific and driven to a specific type of technology. Whereas these two are pretty generic in some way. So we'll talk about more generic methods first to start. So let me take a pause again here. Uh, and see. So now you have multiple methods to explore, right? Uh, many times you are working on variety of organisms. Let's say if you are fortunate, you are working on the human samples in which you have lot of resources available. You have many things which is already done for you. So it's good idea to not to start from scratch, but rather you can utilize some community resource available for you. People are putting so much effort from US various government funding agency side, especially from NIH and various other big projects which are there, which are generating the resources for the whole community. Like CPTAG, they are providing lots of information as a safe portal for many of these peptides for human samples, human proteins of interest, for which what are the possible peptides to select. Again, in the mass spec, as you by now know, that you are looking at everything, you are going to ionize your peptides, and those ionized peptides are going to be read inside the mass analyzer. Not every peptide gets properly fragmented or properly ionized just because of the nature of the hydrophilic composition of that peptide, which sometimes makes them very difficult. So somehow mass spec sees some peptides very frequently, very happily, and some peptides need lots of innovation to do proper ways of either dilation or better ways of ionization. Only then it can be seen. So as a result, you always have certain peptide which can be very easily looked into mass spec for various assays. So for many of those peptides, there are good resources like CP tag where things are already available. You can go and look for your proteins of interest for which peptides, multiple mass spec can actually look at those transitions already and what are the possible collision energy mentioned for them. So that's a good smart way of taking forward. Rather than you optimize everything yourself, why not pick up some settings available from CPTAG? Or another resource is SRM Atlas. You can go to SRM Atlas and look into for which peptides and which proteins. Mostly all the human proteins should be there, but for at least which peptides of your interest data is already available. So then you can pick up those settings and then you can take forward to optimize further from there. Otherwise, you're starting from scratch. So what I am going to share you again the slide is something that how to take it forward from SRM Atlas. So now, first thing is you can search your peptide of interest. You go to the Serum Atlas page, which is maintained by Institute for System Biology, Dr. Rob Morridge and this team, and look into which peptide you are, looking for screening, and most likely for your human peptide of interest, this information should already be available. So now you can choose the right instrument platform for which you want and other parameters, and then you see that what settings you can obtain, which you can just download that directly, and now use this setting in your triple quad instrument to set up the assay. So this can be quick start for doing your target experiment. So how this thing works? So uh, today's session we are split into two parts. Uh, One, we will have the hands-on session about selected reaction monitoring or SRM, and second, we will have the hands-on session for the PRM. So I'll briefly talk to you about what SRM and PRM can do, but then we will have better understanding as we go along. I will have two teaching assistants with me, Sachiran for looking at the SRM workflow and Vipin for the PRM workflow. They will talk to you and show you the workflow with the skyline, how to take it, this kind of analysis forward. A common point of among the two is that you need a good sample, right? And for that, I have another, two Ts, Meher and Shalini, who will show you briefly about the sample prep strategy here. Uh, so let me talk to you again briefly about, just a quick overview for SRM, PRM, and then we will talk to you about the sample pressure part. So now you have identified uh, your peptides of interest. Let's say five peptides you are very keen, and you do not want to from your complex peptide mixture. You are only looking at these five peptides. You want to eliminate everything else out. So you are using a triple quadrupole based mass spec, and you are looking at in the Q1 only those five. You are asking to software to select the precursor for only those five peptides which is of your interest. And now selectively you are fragmenting the most intense ions in the collision cell and generating their fragment ions. This whole thing, which is now you are looking at the Q1 and Q3 pair, is known as a transition, which is unique for a parent and its corresponding daughter ion. So the precursor and the fragment ions constitute that transition, which we say. So for a given peptide, you can analyze more than one transition. Ideally, three transitions is ideal to analyze. And for a given protein, it's a good idea to have three to five peptide at least. So let me talk to you again. Uh, in the Hugo conference again, very recently there was a discussion. Again, these kind of technologies and these approaches are just so relevant and so much latest that I don't think community has still evolved towards one common answer. So one questions are being discussed. Let's say for a given protein, you are selecting five peptides, and for a five peptides, you see that three peptides in your SRM assay are showing upregulation and two peptides are showing downregulation. Now what to do, right? That's one situation. Second situation is all the five peptides are showing upregulation, but the level of upregulation are very different. Like some are showing very high, some are showing subtle change. So again there is no consensus, there is no specific guideline, but although you should definitely read, latest paper on target proteomic guideline published in the MCP, where you should follow up many of the good practices which are mentioned. Uh, but just coming back to this part, which is actually the problem, that you are selecting, let's say, five peptides of a given protein, you are trying to quantify them, but some peptides are showing you high upregulation, some are showing you down regulation. Now, what to trust, what to take this particular information? So then the consensus is that these kind of peptides can or protein you should not report, because in the ideal, uh, depending on which peptide you are selecting, things are changing, right? So it is very difficult until unless that one peptide is so much robust and unique that you see in all 100 patients, and then you are only relying on that one peptide quantification or two peptide quantification. But if you are trying to mix the information, averaging all five, three up and two down, that's not good. Alternate way was, let's say if you are looking at variety of peptides, five peptides, of which three are showing very high upregulation and two are showing low upregulation, that you can still average, because that is still more acceptable. It means that protein is showing you upward trend, but after averaging the whole thing, then you can now get some sort of consensus number. So these are the kind of discussion points which we can discuss as we go along. But main thing to keep in mind is that still there's no clear, very specific guideline, and you have to evolve. So it's a good idea to pick up a couple of peptides, more peptides, and look at their possible 3 to 5 transitions and see how reproducible your SRM assay looks like. Okay, I'll come back again to my. So now let's say the same peptide which you have prepared earlier for discovery work, now you are running and you are running for things here for analyzing in the mass spec instrument, and then you are analyzing that data in the skyline. Again, how this whole thing works, we will talk in some time to you, and let me again talk to you face to face. So the two slides which I flashed very fast is essentially that now you prepared a sample, and those peptide samples you are running in one of the triple quad mass spec to acquire the possible transitions for your peptides of interest, and then you want to analyze the data. So there is a good software available for doing this, which is Skyline software, and that software you can use for analyzing your target data. So this part we are going to talk in much more detail in the hands-on session when session will come. Let me continue briefly about the PRM in the meantime. All right, so in PRM also the sample preparation part or the methodology part remains pretty similar. The sample prep was exactly same. You have done the reduction, alkylation, digestion. You have deserted, quantified, and now you are doing LCMS. Uh, in this case, unlike the triple quad, when we had various quadrupoles, Q1 and Q3 were quadrupole. In parallel action monitoring, we have Q1 as a quadrupole, but Q3 has been changed with the orbit trap. So this is one major change. The mass analyzers are different here, and the quadrupole will work as a mass filter for the precursors. And now the orbit trap, or even you can use even the top for the second analyzer, where the full MS2 scan of the fragment times will take place. So advantage here is, let me talk to you again. So advantage here is uh, that you want to look at all possible peptides and their transitions. Unlike the previous case when you are only looking at selected ones, here you are doing maximum possible both precursor and their fragment time, and then you are doing their parallel reaction monitoring. So this is in some way more powerful because you can do a lot of parallel analysis. In some way it's weak because when you do a lot of peptides and their transitions simultaneously, things may not work out so well because you need to still optimize the condition for each peptide uniquely, which can be better done when you do a SRM kind of a state. So these are pros and cons which we will talk as we go along.